to thank the organizers for the privilege of the floor. I'd also specifically like to thank Dr. Chessie Barinholtz, with whom I work, and who extended me an invitation to participate in this prestigious conference. Today, I'm not specifically going to speak about nanomaterials. We can talk about that another day. But I am going to talk about an emerging area of precision medicine that I think there's a tremendous potential for the use of developing nanomaterials. This involves a new way to think of cancer and involves new ways, we believe, to treat cancer. While I'm going to speak specifically about breast cancer today, I would hope that you would view this as a paradigm which has been explored in some other cancers that we think is probably broadly applicable. So the idea of using bacteria or the influence of bacteria on cancer is not a new one. William Coley from Memorial Sloan Kettering over 100 years ago first expressed an interest on using bacterial products to treat cancer. And I would say in the last several years, there's been a resurgence of interest in the relationship between bacteria and cancer that I will discuss today. So for those who don't know, I'm going to speak about the microbiome, and that signifies an ecological community of microorganisms that share our body. They don't necessarily cause infection, but they can alter physiology. Bacterial cells far outnumber human cells, and this is a belief because of their ability to metabolize and cause inflammation. Other things likely have a great effect on our physiology and the development of disease. And there's a growing body of information on the microbiome based on in the U.S. on the NIH Human Microbiome Project, which has provided a lot of the background for what we are able to do today. So instead of thinking of bacteria as a bad thing or a cause of infection, there's growing belief that there's bacteria in all parts of our body, mostly located in the gastrointestinal tract. But we're also learning it's in many other areas, and as I mentioned before, is having a great effect on physiology. And so we think understanding the relationship of the variety of these bacteria to physiology and disease states has a lot of potential, and the ability to manipulate bacteria and bacterial products may be a window into changing physiology and preventing disease or treating disease. There's been some very interesting work looking using DNA sequencing to define and describe this idea that there are likely microbes in areas of the body that we have traditionally considered to be sterile. And I show you two examples here. One is inside oral epithelial cells, where you see on these images evidence of bacteria. And on the right side of the slide, you see evidence of bacteria within carotid plaques. And this is a completely new idea. Certainly in medical school, we're not taught that there should be bacteria in these areas, nor are we taught that they should affect physiology in any way, but we believe that they likely do. As I mentioned, microbiome is associated with various disease states, and this has been described for diseases like inflammatory bowel disease, type 2 diabetes, and necrotizing enterocolitis, which is a disease which affects newborn babies. And various patterns of the microbiome have been associated with certain disease states, which again suggests the possibility that you can manipulate these populations of bacteria and alter the presence of disease. So there have been described proposed links between the microbiome and cancer, and that's through either the induction of inflammation, which was mentioned in one of the plenary sessions yesterday. Also, there's the possibility of forming genotoxins, which can damage cells. And then finally, there's a lot of interest in how bacteria in certain parts of the body can metabolize hormones or other biologic byproducts and influence the risk for cancer development. There are a number of examples of microbiota-associated cancers. For instance, gastric cancer is now known to be largely driven by a bacteria called H. pylori. Also, gastric lymphoma by H. pylori, and then a number of other organisms have been associated with cancers in different sites of the body. So there's this growing body of evidence that there may be a link between bacterial dysbiosis and cancer development. Well, I'm going to talk about breast cancer, and it's a global health problem. It's the most common cancer among women in the world. The rate is increasing in many parts of the world, which is a significant problem. Germline mutations, such as BRCA1 and BRCA2, and now a growing list of germline mutations, account for only a small percentage of cancers, breast cancers. And I would submit that the cause of most breast cancer is completely unknown, and that's what's prompted us to explore this idea that bacteria may have a role. So how did we get interested in this? It was based on this paper that was published in 2012, which looked at the microbiome of breast milk. And one might suspect that if you look at the microbiome in breast milk, it would either be very similar to the skin of the mother, and the skin of the mother is shown in the orange circles on the slide, or it might be similar to the oral bacteria of the baby, and that's shown in the purple there. But what you see is that the breast milk has a unique microbiome. It's completely separate from the skin or the oral flora of the baby. So this suggests to us the possibility that there might be bacteria within breast tumors that could be associated with breast cancer. So we had a hypothesis, which is that breast tissue and breast cancer have a unique microbiome. The microbiome is different in breast cancer patients and non-breast cancer patients. And we also, because we're a clinical unit, a multidisciplinary clinical unit, can look at things like subtypes of breast cancer and other histologic features of the cancer to see if the bacteria segregate with various types of breast cancer. So we enrolled 57 women. This all has to be done prospectively with cancer, 21 healthy women who were having cosmetic breast surgery. We collected midstream urine samples. We did an oral wash to look to see if the microbiome in distant sites of the body might be associated with the development of breast cancer or the presence of breast cancer. And then very importantly, intraoperatively and aseptically, we collect 2 tissue samples, right and left breast tissue from control patients, and we have tumor and ipsilateral adjacent normal breast cancer tissue from patients having breast cancer surgery. The tissue has to be handled in a meticulous way because obviously there's a lot of potential for environmental contamination, so this is all done in a very controlled and meticulous way. We then extract DNA from breast tissue. We also importantly do environmental controls, and we rinse the pellets using the PowerMag microbiome RNA DNA isolation kit, and then we do 16S sequencing, and then we perform statistics to see if there's associations. So this is a summary of the cohort, 78 patients. We have cancer patients and non-cancer patients. And then immediately after we remove these tumors, they're taken to a hood in the pathology lab, which is adjacent to the operating room, and we collect and quickly freeze the tissue. We collect, importantly, environmental controls to make sure that what we're observing is not just due to the environment. To determine the presence of bacteria, we use a common technique called 16S rRNA DNA sequencing. It's a metagenomic approach. The scheme is shown here. It allows us to analyze tissues and analyze a wide variety of bacteria within these samples at very low levels. This is a quick slide showing the characteristics of the patient. These are typical breast cancer patients, and the demographics are shown here. This only becomes important later when you want to do analyses for factors associated with differences in the microbiome. And then also a bunch of other clinical factors showing a distribution of patients that we were able to analyze in this study. So this shows, these are principal coordinate analysis showing the analysis of the overall populations of bacteria. Shown in panel A shows the statistical difference within the breast tumors themselves comparing cancer and non-cancer patients. The cancer patients are shown in yellow and the non-cancer patients are shown in blue. We looked at other factors like in panel B, that's alcohol consumption. We look at tumor grade, also significantly different when you segregate the tumors by grade and look at the presence of bacteria. We also importantly looked at lymphovascular invasion. That's shown in panel D, and again you see a statistical separation in the principal coordinate analysis between patients with lymphovascular invasion and without. And we also see a segregation in panel E for patients with and without HER2 positive breast cancer. So a lot of factors associated with differences in the microbiome that really need to be further explored. So we have determined that the breast cancer is associated with a unique microbiome in humans. The breast tissue microbiomes clustered significantly different from non-cancer patients, and this is driven by relative abundance of Methylobacterium, a specific bacteria in cancer patients. And we also saw different bacteria, Alcaligenaceae, which are increased in breast cancer tissue compared to control tissue. Interestingly, also for the first time, we showed significant differences in the urinary microbiome of patients with and without cancer, which has not been described before. These are all asymptomatic patients. They had not received prior antibiotics, and we're seeing differences in the urinary microbiome, which could be a source for a screening test in the future for breast or other types of cancers. Our observations suggest that local breast microbiota in cancer and urine differ in patients with and without breast cancer. This is a schematic showing differences and shows you that the methylobacterium shown on the panel A is decreased in tumor patients compared to non-cancer patients, and you also see in panel B an increase in alkalogen which is increased in breast cancer patients. This is a plot which looks at the distribution, a striking distribution, and differences in the urinary microbiome of patients with and without cancer. You see on the left side the patients with cancer, either peri or postmenopausal or premenopausal, and you compare that to the patients on the right side and the non-cancer who are peri and postmenopausal, and you see strikingly significant differences there. So in conclusion, we think that breast tissue and breast cancer are significantly different compared to environmental controls, so we think there is a unique microbiome. It's different in cancer patients and non-cancer patients. We think specific subtypes of breast cancer segregate with the microbiome, and certainly we found for the first time that urine has an increase in gram-positive bacteria, which is really still unexplained. So we're not the only study to look at this. We think ours is the most carefully done and best study, and I'll explain why. But these are the other 4 or 5 studies which have looked at the microbiome in breast tissue of patients with breast cancer. Most of these studies suffered from 2 problems, one of which is they tried to use paraffin-embedded tissue, which was not specifically collected for the purposes of analyzing the microbiome, which could introduce a serious source of contamination. And most of these studies have not looked at the tumor tissue itself. They've looked at surrounding breast tissue. So this is an area in evolution, but one, again, I think you should be aware of. So what are the opportunities for exploiting intratumoral microbiome? We're beginning to ask questions, does intratumoral bacteria cause or promote breast cancer growth or spread? And then a more intriguing question that we're starting to work on is, are intratumoral bacteria associated with chemotherapy resistance? And let me just mention that briefly at the end. There's this recent paper published from a multidisciplinary group led by the group at the Weizmann Institute, published in Science, which looked at patients with pancreatic cancer. And in this paper, they describe a microbiome within pancreatic cancer. They then also describe treating the tumors with antibiotics, and they show that the use of antibiotics can break resistance of the tumor to gemcitabine. And they postulate that the effect is that the bacteria that are present within the tumor have chemistry which can inactivate gemcitabine and make it ineffective. So we're now working on this idea, can you destroy intratumoral bacteria and make our traditional treatments more effective? And this is an area where, again, I think nanomedicine has a real role. This is a thank you to our entire large group, which has made all this work possible. Thank you.
Wow, very interesting, very interesting. Any questions? Well, I have one. Is there any idea how these bacteria get there?
We're working on that. So it's not really clear how they get there and what they're doing in sterile parts of the body, how they get there, we don't know. One of the ways we're attacking this is through some early phase intervention studies where we actually give probiotics to patients with newly diagnosed cancer, and then we're analyzing the tumors to see if there perhaps is a relationship between the oral intake of certain bacteria and adjustments of the microbiome. But it's not clear. I mean, there was a theory postulated yesterday, maybe some believe that it comes actually from the breastfeeding and the breast milk, and that may lead to lifelong colonization, that kind of thing.
And there are different species in different, I think, parts of the body, right?
Yes.
Wow, okay. Oh, yeah, I'm sorry.
Hi, thank you so much for the presentation. So how would you define this PGP or P-glycoprotein efflux pump? Can be expressed by some stem cells or just initially activated drug-resistant cells in the breast tumor already there, or just acquired, versus the bacterial resistant in breast stroma?
That's a difficult question. Yeah, that's something that we obviously needs to be looked into to figure out how we break the resistance.
Okay.
Thank you.
Oh, and any more questions? Well, if that's not the case, thanks a lot, and good luck with this research, which is kind of unique.